Good afternoon, everyone. Hope the echo is gone. Yeah, it's gone. Okay. Welcome to the new NITheCS Colloquium series. Yeah, this is the first talk of 2024. So happy New Year, if it's not too late. Yeah, hopefully it will be a very productive year for everybody. So I'm very happy this afternoon to be able to introduce you you can already see Dr. Ruan Veldtman. Ruan did his PhD at the University of Pretoria. And then I just can't read it off from here to training my memory. Yeah. After which he joined the South African National Biodiversity Institute, but he's also associated with the Department of Conservation, Ecology, and Entomology at Stellenbosch University. Yeah. And Ruan, thank you very much. This is load shedding, but we have a UPS this year, which we didn't have last year. So there are steady, constant improvements. Yeah. No, I'm very grateful for your talk, and in particular, I'm very grateful to your title, because that will set the scene for interesting titles for the year. Yeah. So thank you very much, Ruan.
Thank you very much, Francisco.
And welcome to start with your talk.
Thank you. Thank you for the introduction. Hi, everyone. So my talk today is going to be about research that I've been doing with colleagues on the German wasp, which is an invasive species in South Africa. And this is basically a history from when we started the work right to what's happening currently. And it's one of my really favorite projects that I'm busy with. So yes, without further ado, to introduce the species. So Vespula germanica is a eusocial wasp species, which means they form a social colony where you have workers and a queen that produces new workers, and basically it functions as a superorganism. And this is how you have to also think about the species. So it's, you have individuals, but they together form a colony, and the colony is then a superorganism. So we have two invasive social wasps in South Africa, both only found in the Western Cape. The first is the paper wasp, which is black and yellow as well. And the species I'm talking about today principally is the German wasp. So you can see to the untrained eye, they look quite similar. But in the talk today, I'll show you the differences and how interesting the two, especially the German wasp, actually is. These wasps have stings, and different from honeybees, they can sting multiple times. Here we have the sting dissected out of the insect, and you can see the venom sac. You can see the venom sac there, and there's the stinger. And if one's not careful, you can get stung. And this just reminds you to be extra careful. So this was actually through my protective gear, and my face just slightly touched the mesh, and I got biffed in the eye. But there are fancier ways to measure the aggression of colonies. So for example, here we have a device which emits alarm pheromone, and then the wasps are triggered to attack, and they can measure the force of the attack with this device. So just these two species, I said they look quite similar, but there are characters if you zoom in and focus a bit. So what we always say to the public, if you see one with black antennae, that's the German wasp. So if you look there, whereas the paper wasp has orange antennae. And if you look for a while, you can see that the abdomen, this one has more almost arrow patterns, whereas this one is more a just variation in the color. Okay, and I haven't actually made this poster formally to be circulated, but this is on my idea board. So if I explain to people the German wasp, it flies with its legs pulled up, but with the paper wasp, it flies with its legs hanging. And also the kind of flight differs. So if you see something buzzing around in a bee-like manner, that'll be the German wasp. But if it's in a slow, lazy flight, that would be the paper wasp. And then the drawings here at the top. So for the German wasp, they sometimes nest in tree hollows. They could also be at the base of a tree or on the ground. in vines, they would normally be then in the rows, and in your buildings, they could go into artificial holes like ventilation holes. Whereas in contrast, the paper wasp, the nest would be on the margin, would be under the roof or on the vine. But basically, these ones like heat and sun, and this one likes shade and cooler conditions. Okay, so when we explain anything to the public, it is important to make sure we get the right identifications, because otherwise you get false reports. So in many cases, we hear, oh, we've got German wasps. They send a picture and then we see, okay, it's actually the paper wasp and not the German wasp. Here we have a native paper wasp. There's a potter wasp. There's the honeybee, a pollen wasp, a sand wasp. So you can see there's a lot of entomology in this as well. Okay, so just the principal difference with the paper wasp is that they build a nest, which you can see actually the nest cells. Here's another example. And basically in the social evolution of the species, this is a less evolved species. So it has a single nest comb. But we also have workers, but there's not a big distinction in their morphology. Here again, we have a native paper wasp. This is Rhipolidia species. And it's again similar. We have wasps guarding the larvae. And they guard the larvae, and we have a queen that lays the eggs. Here we have a hornet. So we have no hornets up to date in South Africa. And these are larger insects. You can see the size here compared to the western honeybee. You see it's almost double. So hornets, and especially this species, the Asian hornet, which is now invasive in Europe, is a very ruthless honeybee predator. And basically, they have evolved to eat honeybees. Then we have another wasp that's also thankfully not in South Africa. This is colloquially known as the giant murder hornet, or Vespa maderina, and this is the largest social wasp that one finds. Quite venomous. About 12 stings is fatal to a normal human being. So when we talk about the German wasp and we're looking for noting their distribution, basically we have to find this, and all you'd see are these holes. And if you look carefully, you can see the wasps flying in and out. And then they've nested behind this rockery. So if we excavate the soil, this is basically what's behind. And as I showed with the paper wasp, we have multiple levels. So the paper wasp has only one layer, and it's also not covered in a larger structure. But with the German wasp, we can have up to 11 to 13 levels. And basically, food comes in at the top, the wasps crawl around the outside, and at the bottom is the entrance. And the most important people live here at the bottom, and the more expendable ones are at the top. So you see another diagram just to show what I talked about. And then basically it starts with a single little cell. And this is a very important time because the queen needs to produce enough workers that can take over from her before she runs out of energy. So there's basically a timer. She's doing it against the clock. And if she runs out of energy or the conditions aren't favorable, that queen will die, which means you won't get a colony. Just want to show this video. Sorry, so practice this. Francesco, do you know if one plays a video, where to press? Oh, just on the video. It is actually nice to see. Let's show this one. There we go. Okay, let's skip this one. So basically here, thanks, Francesco. Here we have a video that's not working, but basically this is a German wasp nest in the roof of a lapa. Here you can see the thatch. Sorry, it's on its side. When it plays, it looks correctly. And then you can see the wasps flying around. And this silvery structure is actually chewed wood, which the wasps then build in this structure, and the combs are inside. Here we have a very special nest. That's the picture was courtesy of one of my collaborators, Edwin. And this is a very weird nest in that we have a hole here and also here, and then lots of other funny aberrations compared to that smooth nest I showed. And let's hope this one works. Doesn't want to work. Okay, now we'll just carry on. Thank you. Okay, so the previous video was a nest removed by a wasp controller, Edwin, who's not pictured, and it was a massive nest. And instead of being underground or in a wall cavity, it was actually completely externally to the structure. And he removed it without any safety clothes. And I asked him how many times he got stung, and he said only once behind the head, but I think once is too many. And I showed this video, which was received with great amusement by a high school here in Stellenbosch. And it was a nice way to enthuse these young people to look out for these wasps and make them interested in the natural world. And when I give these talks, I always show the slide of iNaturalists to try and encourage people to, when you see something interesting, an animal or an insect, take a picture, put it on iNaturalist, because this is data that's being captured and you can be part of citizen science. So here we have some pictures of wasp removal. So here are two field workers gearing up. Here they're in action, busy vacuuming wasp from their nest entrance. The wasps are in that little drainage hole. And here we have my PhD student, Jacoby, busy digging out wasp nest after it's been doomed, so you don't have any incidents. Then we also came up with a very nice method of actually capturing the workers. So the queen would build a nest. It would look like this. Then you could take the equipment, put the end of the apparatus at the entrance. The wasp would then be sucked up through with this petrol-driven blower vacuum. Here you can see the equipment. And the wasp would then accumulate in this box, and then you can put it on dry ice or manipulate it. And then basically you would have all the foragers that were flying around captured neatly in a box. So this is currently in press. And we hope this will also help other people working on these types of social wasps. And then down the pipeline, you could look at genetics of the wasps, or you could do measurements or look at prey items like my PhD student is doing. Okay, and then a family is very useful. Here you can see my children helping me create this figure. So here we have all the different components of the vacuum. And to get a nice view, I had to put one of them on my shoulders. So where do we start with this story of the German wasp in South Africa? So firstly, in 2012, we actually put in a proposal, which was then funded by SANBI and the Department of Expanded Public Works. Pictured here, back in the day, it was as it was known. And we advertised for 4 MScs and 1 PhD in this lot, and then again later a postdoc and another project. And basically, this was our first announcement to the world that we are actually working on these wasps. Then each year, we basically had anecdotal reports. Here we clearing a wasp nest from someone's house right next to their driveway. Here's another interesting nest that was inside on a farm, inside a piggery on the side, a very menacing looking nest. Then in 2014, we helped the municipality train a few people how to do wasp removals. And then things really gathered momentum in 2016 when we had Dr. Derek Daly volunteer his services. So Derek is a retired behavioral ecologist and geneticist from the University of Liverpool. And his perspectives are very useful because he lives where these wasps are native. And we work on these wasps where they are invasive. So he's really helped with the research effort. So yeah, one of the ideas he had, you see this little orange blip. This is a German wasp that has been covered in fluorescent powder and then released. And the idea is that you would see where it flies to and then try and discover its nest. Here in 2018, sorry. Here in 2018, we have Derek very proud because he's finally found a nest we basically looked for a week in Stellenbosch Gardens. And it was just there right near the visitor's entrance. And then through the years, so Derek, another 3 years, he's visited us. And each time we have different insights and scientific exchange. So it's a very valuable his collaboration. And if you then map what we did over time, you can see here the blue line is the number of wasp nests that we've discovered and that goes into the database. You see if we have a good year, it runs at about 100. But the point is if you add all of those over time, we have a very large number of wasp nests that are being documented. And then you can start to look at patterns. Because one has to remember, or I just want to say, so you've seen the line, and then in the background, this is actually the information. So here we have, in Google Earth, each nest is represented by a line, and it's got a label. So basically, this is the background of the data. If we want to find a nest in a specific area, we can zoom in and get it. So it's important to know exactly where those nests are located. And here I want to show quickly a map of a current distribution of the German wasp in the neighborhood where I stay. So each yellow star is a nest. And you can see here it occurs right outside someone's garden, here a whole cluster in a park, and here in a different part of the park. But what I want you to think about is that around each nest, if you draw a 200-meter radius, this would be the normal distance or the maximum distance that these wasps fly around and look for food. And then for each of these nests, we can draw a circle. So also spatially, we can now think of the superorganism and how it interacts with the environment. So not just at the point, but in the immediate vicinity of that nest. Yes, so that brings me to the social wasp invasion team. It doesn't have such a formal name, but just to list everyone. So I've talked a lot about Dr. Derek Daly from the UK. Then we have Dr. Gulu Bakker, who lends his geographic and GIS skills and helps us map some of the patterns we see. Dr. Barbara van Asch is a geneticist. Prof. Adrian van Niekerk works in the Department of Geography, and he's helped us with accessing data from there. Prof. Kang Wee is a mathematical mathematic ecologist. So you can see a whole range of skills that is available and working with these different colleagues. Then there's also 2 PhD students that are currently busy. We have a postdoc, Dr. Damien Gilgon, and then our special friends, Andres and Maiti from Argentina. And Argentina is really interesting because geographically, we have a very small German wasp invasion, but they have one that spans several degrees of latitude. So it's really interesting to work with them and see if we repeat our work in Argentina, do we see if there's any general patterns that one can then use to manage the species or understand the species. And then last but not least, Marco and Edwin. So Marco works for the City of Cape Town's invasive unit, and they deal with the public who reports nests. And over the years, we've probably received more than 75 nest records from Marco alone. And then Edwin is a collaborator. He does snake removals, hence he's not scared of wasps if you're dealing with snakes. And he's also probably by now contributed more than 50 nests. So this is a large component of the work. So working with multiple collaborators in multiple disciplines, and then also with people that interact with actually the practicalities as well. So I'm just quickly, as the more scientific part of this presentation, going to share with you a presentation I did at the Entomological Congress in Stellenbosch that was in the winter of last year. I've introduced some of the co-authors already, and the idea was to understand why is the German wasp not that successful? Why is it not spreading as fast as you see in other countries? And specifically, we're seeing the Western Cape as an example of a Mediterranean type invasion. So globally, social wasps rank as some of the worst invasive species because they can have ecological and socioeconomic impacts. And as part of the scientists working on that, we're trying to work out what is the likely routes of dispersion, what are potential sources, so we can gear programs to stop invasions before they start, because it's much cheaper and easier to prevent the invasion than to deal with the invasion when it's really bad. And then the whole idea behind this paper was to use genetic data to understand what is happening with this invasion if we then combine the genetic data with the satellite data, which helps us map with remote sensing, the different climatic variables that are potentially important to these wasps. So just as an introduction, if we look at the macroecology, in the red circle we have where we think our German wasps have originated from in South Africa, but we are still to confirm this with genetic work. And then you can see on basically all other continents, we have invasions of the species. So only Antarctica has no records. And then in South Africa, the normal way how you would try and estimate the distribution of invasive species is to do some climatic modeling, but this is at a coarser scale. So if we look at South Africa, the red areas are highly suitable, whereas gray are completely unsuitable. And this was a climatic envelope that was already produced, but we refined this by adding irrigation as another factor. And using both Argentina and Australia data, we actually saw that we had improved success in explaining where we find these wasps. But what was still a puzzle, here in the Southern Cape and also the rest of South Africa, we have massive areas that is very suitable for the wasp, but we don't find them there. They are still only found in this very special part of the Western Cape. So then we started to look at various models and approaches to understand the invasion. So this is a hotspot map, and basically it shows the values that are low, those are in blue, or values that are very high, that are close in proximity. So what that means, in this blue area, we have low summer maximum temperature for all these nests that are found here. Where if we go more north into Wellington and Franschhoek, we see there's mostly high summer max temperatures. But it's very interesting how we go from cold to neutral to almost hot, high to very high. So there's definitely spatial structure. And if we look at rainfall, mean annual rainfall, we see the same thing. now high values of rainfalls in red, and we can see in the mountains we have rainfall. As we go out Stellenbosch, then it starts to be low values. So again, there's definitely something going on with these different climatic variables, because we're trying to explain the distribution more finely than we can do with the climates model. So this was also a bit of a breakthrough, was to look at the normalized difference moisture index. And this we got Dr. Gulubeka's help to look at. So here the black dots are actually Vespula germanica nests. The blue is incredibly dry environments, whereas the red is high shade or high moisture availability. So, and the blue lines is the East River. So basically you have forested areas, planted forest, which produces shade. You have lots of trees along the river producing shade. But if you go up the mountain, the natural fynbos, that would be very dry environments. And what we see is that our wasp nests principally follow this sort of edge where we have on the edge of high moisture environments. So this was really interesting. And if we zoom out, again, the same thing, red is high moisture and blue is dry. We can see at a large scale, the potential suitable habitat for these wasps are very fragmented, and a large portion of it is blue. So now the wasps have to move from one red area to another red area, and this is what we think is slowing their invasion as well. And there's also other factors. So we have lots of humans in these areas, but in this area as well, there's also high population density, but we don't see the wasps. So there's lots of factors at play here. But we do see, since we started the project, invasions in each of these different wind directions. So the wasps are still slowly, albeit slowly, they are still spreading further in this part of the Western Cape. Okay, so the aims of this work, of this specific talk, was to compare the distribution of the two haplotypes we found in the Western Cape, and then to determine if these haplotypes, so if you sample a wasp, you know it's a particular haplotype, that nest, all the individuals in that nest is of that haplotype. So when I refer to haplotypes, I actually mean the nest is of a particular haplotype. And then what we wanted to do is, if we map those nests, can we see any patterns in environmental variables that we measure? And then to see if we can then also explain different rates of invasion linked to haplotype. Here are some of the environmental variables we use. So distance to a main river course. So the river would have water, but also shady vegetation, which would both be advantageous to the wasp. High mean annual rainfall would be preferable, and then other variables linked to temperature. And we got this information from the Center of Geographical Analysis at Stellenbosch University. So here I plot the distribution of NDMI values. And basically you can see where we find the wasp nests, the values are offset to the right, which means the wasps tend to be in areas where the NDMI values are higher. So we did some work. If you compare where they're absent to a area around the nest compared to where the nests are exactly, we see the whole time we have an increase in NDMI as you zoom into the wasp's nest location. Then in terms of the genetics, what we did was to actually redo some work and expand the part of the mitochondrial genome that was looked at. And instead of looking at only about 1,500 base pairs, we looked at a whole range of genes, basically making sure we'll pick up any variation that's there. And we also had a few samples from other countries, which also helped us make sure that these would be useful for other people. And basically, previous work showed that there were 6 haplotypes, but when we redid the work, we find only 2 haplotypes, so a common haplotype, which will always be red in the rest of the talk, and a blue haplotype that would be more rare. And these others, we couldn't, using the same samples, we couldn't replicate. So these are sequencing errors. Okay, so if we map these nests on the map, we see a very interesting pattern. So our blue haplotype, or the rarer haplotype, is mostly on the Cape Peninsula around Table Mountain, with these odd nests. If you zoom in, this would be the Eerste River, and then all the other nests that we sampled across the Boland and the outlying areas would all be the common haplotype. So definitely there's a very, very strong genetic signal in the spatial distribution we see. And then our goal of mapping this to variables. So here we have mean annual rainfall. And if we look at the different nests sampled, there's no statistical difference between the two haplotypes. Again, if we look at how far they are from a river course, so this would be 1 kilometer or 1,000 meters. So also no difference. But if we then look at mean annual maximum summer temperature and also mean annual maximum temperature, so summer maximum would be the 3 hottest months during the year. So this would be from January to March. And then we suddenly see a difference between the haplotypes. And this is quite significant if we have 2 degrees difference in the mean annual temperature for these species. And again, we see it also for mean annual maximum temperature, but not for any of the other temperature variables. So we can conclude that summer maximum temperature seems to be a very important driver of where we find the different haplotypes, seeing as that all the other variables were not significant. And this supports our hypothesis that we have had 2 invasions and that the second invasion was by haplotype more suited to South Africa, and it's therefore more common. And it seems to do quite well in our Mediterranean climate. So now we're busy with future work to see if we can see if there's any physiological differences in terms of the queen's ability to survive during with temperature exposure, and also if transported by humans in cars or trucks, and see if we can link this to haplotype. And we hypothesize again that the hot haplotype is better able to survive these jumps that people cause by moving goods by accident. And then if we look at the invasion history, so Vespula germanica, all Vespula germanica was concentrated within this area up until 2000. And then between 2000 and 2005, suddenly we had a explosion in new localities where the German wasp was found. And so basically we have a pre-2000 and a post-2000 distribution patterns. And we think this is due to the different haplotypes. So if you plot localities where we think you find the cool haplotype, we map that over time, and the distance of the kilometer distance by road from the original locality, which would be Stellenbosch. And for the common haplotype, we think it is Stellenbosch, based on the spatial patterns. We can see that the rate of invasion is much slower for that first haplotype compared to the second haplotype, which is quite fast. But by international standards, 30 to 60 kilometers spread per year is quite normal for these wasps, where the habitat is really suitable. But South Africa, as I showed with that very fragmented suitable habitat, the invasion is slow. But even within that, we can map 2 different invasion speeds if we use the genetic data. Okay, so then to conclude, the first invasion we hypothesized was by a haplotype coming from a cooler European climate. And basically the wasp was trapped around the Cape Peninsula for all those years. And then around after 2000, we had a second invasion by a queen that originated from a more Mediterranean climate. And because the Western Cape is also Mediterranean, this has allowed the wasp to become more successful when invading. And our data suggests that invasion will still happen at this rate of 6 kilometers per year. But of course, if we have a massive jump dispersal, this will throw out those figures. But on average, this is what we're seeing. So, and because it's difficult to actually work out this accidental dispersal, that's why we work with Kang Hui, so we can try and build models where we can look at the more stochastic process of these jump dispersals. Okay, so we think the invasion of South Africa was the slowest in the world because our first haplotype was not well suited. And only very recently, or much more recently, we had a second invasion. And this invasion wasn't as slow, but if you look across the whole time that South Africa has had this wasp, it looks really slow and it's odd. And we think this whole production line of different fields looking at these different patterns, trying to explain them and using different filters helped us explain this interesting pattern. Okay, so what we're planning in the future is a global analysis of haplotype distribution for this species. And to do this, we will cooperate with lots of countries. I've shown, so actually this is the distribution of all Vespula in the world. North America is really interesting. They have a lot of native Vespula wasps, several species, but they also have germanica, and then also Australia, New Zealand, Argentina, and then also on many of the islands. So our plan is to host this International Wasp Symposium to encourage colleagues to visit us and share their findings, and then probably do a global analysis by combining everyone's samples, and then hopefully we can see where our actual haplotype originates from, and maybe other countries as well which have a puzzle about where their invasion originates from. So I would just like to acknowledge several people, and this is on top of all my collaborators that I've had many fruitful discussion with, my family, which often helps me in the background, and the wife that doesn't complain if there's wasp samples in the fridge. And then also to our colleagues, so Marco and Heine from the city of Cape Town, which helps with getting data from that area. Edwin getting data from other areas. Babylon's Touren, who's helped sponsor Derek. And then also just the landowners and the public for noting these records and feeding it to us, which we then can roll out into our scientific effort. And then just a thank you to the Department of Forestry, Fisheries and the Environment for funding my salary. And yes, if you have any questions, please ask. I'd be interested to know. And there's my email address. Thank you very much.
Thank you very much for a very exciting talk. I'm sure you'll have plenty of questions. Any questions from, let's start with the physical audience, and then we can ask the online audience if they have a question as well. Who wants to go first? Here we go.
How much of your work is activist, so removing wasp nests? Because I can imagine if you're removing wasp nests, then you're also interfering with your data about trying to predict how fast they're spreading and those sorts of things. So how do you balance like the prediction versus the mitigation, which is the ultimate point, right?
Okay, no, thank you for that question. So to say the nest I've shown, that is not, I think there's many wasp nests that we're still not sampling. So even if we kill all the nests we found now, there's still enough to actually carry on the invasion. But even if that nest is destroyed, if it goes into the database, we have all the spatial information, we can do a retrospective analysis of what's happened. And each year is a new year. So a nest would produce new queens, they would fly, they would disperse and build their own nest. And depending at what time of the year that nest is actually destroyed, the queens may have already dispersed. So the short answer is that even with the removal, we can still do our work. It's not a problem. And it's a way to engage the public and get feedback from them and encourage people to put their records on iNaturalist. So there's still work to do to formalize that actual data capture from the public.
Thank you, Ruan. So I have two questions. The first one is, if you look at the range expansions in the southern hemisphere, you normally see a vertical expansion, so along the latitudinal gradient. And in South Africa, we don't have that very strong latitudinal gradient. Could that explain why in South Africa, it's not just for the German wasps, but for many invasive species, we have experienced a very slow range expansion compared to other places.
Yeah.
The second question is related to, do we have a candidate biocontrol agent for this species?
Okay. Thanks, Gang. So your first question, sorry, Gang, just repeat quickly the first question.
So it's, the slow range expansion in South Africa.
Thank you. Thank you. So I think what's happening, a lot of European-like species that have a history of being transported to South Africa, they would be in the same boat of this wasp in terms of being from an area that's normally cooler than you would find in South Africa. South Africa is a hot country. And because our latitude runs out at around 34 degrees, I always say jokingly to the Argentina, you're so lucky, your country is so big, you can still have invasion going down south. And I think if our country looked different and there was more space going south, you would have seen invasion further, as you see in other southern hemisphere countries. So I think it's because we have a hard limit to go north. And it's interesting, Africa also doesn't have hornets. So there's definitely larger patterns at play, evolutionary and also the glaciation and those things. All of those we still need to look at. But this very slow invasion of Vespula germanica has given us the opportunity to really understand and come up with ideas of what's happened. And then we'll see if we can find evidence that this is also happening in other continents. And then hopefully we can one day crack this nut of what limits social wasp invasion, because then you could also be more prepared for future species. And then your question about biocontrol. So I'm aware of 3 agents. New Zealand in the 80s tried a ichneumoniid wasp. It wasn't terribly successful, but I think they said the major drawback of that first work was that they had a population from Switzerland, and I think that wasn't very suited to the wasps they had. They thought they'd have their wasp from the UK and those areas. So they busy with, it's Bob Brown at Landcare New Zealand. He's busy now with a trial and he's reported that biocontrol agent from the UK, and he's going to test it now and see if there's it improves. And then we have a fly that attacks the colonies, and then also there's a beetle. But again, I don't think we can expect them to have a massive impact. It would be very it would be a specific nest, and maybe the nest right next to it wouldn't even notice that agent. I mean, a biological control agent or that species would go extinct if it was very adept at really hammering its host. So I think in most cases they have a minimal impact on the actual species.
Thank you. Any more questions from people in the audience here? No, then I think we have a few questions from our online
Hello, can you hear me?
participants. And Donovan, will you read the questions, please?
I'll read them, yeah. Okay, so the first one says, how have you addressed undersampling biases in your study, and in what ways have you integrated the temporal aspect of invasive species spread into your approach? Specifically considering that time is a significant constraint on the realized niche of invasive species, indicating that absence in an area might not reflect unsuitable environmental conditions, but rather insufficient time for dispersion or spread?
So the data I've shown isn't from specific years. There's definitely not the resources to have a formal survey each year to map where all the nests are. So it's a slow build over time, sampling the different areas. And what we also do is to try and cover areas that haven't been covered before. And our solution for correcting this temporal bias is to actually build a probability model, which we're also busy with, that will link the geographic information we're using to come up with a probability map. Then we can go to those areas and verify that there is indeed an absence or confirm it as a presence that we didn't know about. And that'll give us some idea of how accurate our work is thus far. But I can tell you when we go to a new area, you see a few elements in the environment, it looks like wasp habitat. And then in more cases that we are right. But yes, it would be a luxury to have a very nice temporal data set, but yeah, it's too difficult to collect, but we are devising a way of addressing that weakness.
Okay, and then there's one more. This question says, I just wanted to know, are these wasps a threat to our honeybees? And how do I determine if it is in fact the Vespula germanica? We have yellow jackets in our garden in Durbanville.
Okay. So short answer, Jacoby's work is showing that the wasps take about 10% of their diet is honeybees. So they're not a major predator, but I wouldn't say it's insignificant. But it's not at all sites. It's also at specific sites. What is frightening, if we think that's already 10%, if we would get that Asian hornet, that would be catastrophic. We'd probably lose about 50%, or 50% of what that wasp eat would be honeybees. And compared to other parts of the world, we have a very large wild honeybee population, and it would be very difficult to help control the wasp where they live. Whereas in Europe, a lot of the colonies are managed, so it's very easy to come up with protocols. Sorry, just repeat that second part.
How do you know if it is Vespula germanica?
Right.
So this person has some yellow jackets in their garden, so they want to know if it could be that, I guess.
If they can take a picture, they can submit it on iNaturalist. And expert or myself, I have a project that filters data specifically for those species. And we'll have a look. And usually it's if there's a nice picture with of a few key structures of the individual, you'll be able to say, okay, it's a paper wasp or it's the German wasp. And because we only have those two invasive social wasps in South Africa that are black and yellow, it's quite easy to say. And Durbanville, it depends where in Durbanville. Most of Durbanville has very high density of the paper wasp, but around Tygerberg, there is definitely a stronghold of the German wasp. So also if I get the GPS record, I'll be able to also revise my, to say what I think it is. But a picture is key.
Excellent. Here, one more question from here.
Thanks. So if we look at the structure of the nest, the location of the nest, as well as its internal construction, can we learn anything about how they adapt to the climatic envelope at which they are being found? Thanks.
That's a very good question. We think definitely these wasps are resourceful. And what you normally see with invasive species is that they would invade in a new environment, and the invasion would at first be slow, and then over time they start to adapt to it. And we think those nests with the very peculiar structures is an adaptation to South Africa's warm climate, and we think that's a way of cooling the nests. So we're also busy with work to actually see if we can prove this theory of ours that the wasps are able to build their nest in such a structure that it helps with the cooling, so it can ameliorate the effect of the climate that's actually quite unsuitable for them. And then remember the queen as well. She's been selected to build a nest in a spot where that she has a high chance of producing offspring. So the queens that get it wrong, the queens that go for a too dry environment or build the nest in spring, where that spot in summer is very hot, that colony is going to die if they're not able to cool the nest. So the wasp can also collect water, but the more water you collect, you're wasting energy because you're not bringing in food, you're bringing in water. So it's a whole time it's a balancing act. But definitely we think the organism, social wasps are plastic. There is some behavioral things they can do and structural changes that can help them survive.
No more questions? Then Ruan, it's my big privilege to thank you for
Thank
the
you very
very
much.
nice talk and to offer you a
Ah.
little magazine or journal that NITheCS sponsors. Yeah?
Fantastic.
And it's intended to encourage young and not so young people to get interested in what NITheCS supports.
Okay, thank you very much.
Thank you.
Thank you, everyone.
Very much.